Hello, very good morning to all of you. Uh, our guest speaker, Professor Astush Kumar sir, are connected with us. So, Astush sir, am I audible?
Yes, you are.
Yeah, yeah. Thank you, sir. Thank you. Nitin sir, please.
Good morning, sir. Good morning, all of you. And as you know, today is IIMT University organizing the 2 days international conference on the topic current trends and recent development in pharmaceutical sciences. So I welcome to all audience and all speakers present here. And I special thanks to attend this conference for our honorable guest, Professor Asutosh Kumar, as a speaker from IIT Bombay, and Professor Javed Ali from Jamia Hamdan, and Dr. Yasir from Ethiopia, Asela, from the University of Health Science, Ethiopia, Asela. I also thanks to Dr. Anuj Mittal, Dean Pharmacy, College of Medical Science, IIMT University, and invited him for all the speakers. Thank you, Dr. Anuj.
Thank you, Nitin.
Very good morning to all of you and wishing you a happy new year 2022. As we all know that today IIT University is organizing 2 days international conference on current trend and recent development in pharmaceutical sciences. I am heartily thankful to our chief patron, Shri Yogesh Mohapatra, Honorable Chancellor, and Dr. Mayank Agarwal, Honorable and Managing Director of the University, for providing the such platform to organize a wonderful international conference. Our patron, Dr. H.N. Singh, Honorable Vice Chancellor, co-patron, Dr. Deepa Sharma and Dr. Satish Bansal, Pro Vice Chancellor, and my team for organizing this international conference. I also welcome Dr. Professor Ashutosh Kumar from IIT Mumbai, Professor Javed Ali sir from Jamia Hamdan, and Dr. Mohammad Yasir from Ethiopia. I think this conference is a platform to enrich our knowledge over the current trends that are going under to the pharmaceutical sciences, and hopefully that it will be beneficial for all participants. And I am thankful for all the eminent professors and personalities for sharing their knowledge on this platform. Thank you very much. Thank you, all of you.
Thank you, Anuj sir. Now I would like to invite Mr. Atul Pratap sir for sharing the achievements and credential of Dr. Asutosh sir. Thank you.
Thank you, Nitin sir, for giving this opportunity to introduce our guest speaker, Professor Astush Kumar. Professor Astush Kumar is working as a professor in the Department of Bioscience and Bioengineering, IIT Bombay, Mumbai. His work involves studying the structures and mechanism of action of protein complexes such as SCF complex, specialized nucleosome, and amyloid fibrils using state-of-art solid state and solution state and MR spectroscopy along with other biophysical tools. Astos has made significant contribution in developing NMR method and applying those on complex biological macromolecules to reduce the structure and dynamic of these molecules. In the same time, he started working in the field of biopharmaceutical, where he developed novel NMR method to investigate HOS of biosimilars and biologics in their formulation state. Professor Kumar received his MSc in chemistry from IIT Bombay, followed by his PhD at the Tata Institute of Fundamental Research, Mumbai, supervised by Professor R.V. Husser in 2007. He did the postdoctoral research work under the supervision of Professor Mark Balges, Professor Adam Lang, and Professor C. Brisinger at MPI for Biophysical Chemistry, Göttingen, and Germany before coming back to IIT Bombay. So this is the brief introduction for our guest speaker. Now hand over to Dr. Nitin sir, please.
Sir, Professor Asutosh sir is invited to deliver their talk on the topic relevant to our field. So welcome, sir, please.
Okay, thank you very much for inviting. So is audience offline or they are going to join now?
YouTube live, sir. The audience is YouTube live.
Okay, fine. So what I'm going to do is sharing my screen. Is it visible?
Yes, sir, visible.
Good. Okay. But how are they going to ask questions then?
I will take a question from the YouTube.
Yes, I will take the question from YouTube chat box and ask at the last of your session.
Okay, no problem. Fine. So yeah, very good morning to all of you and Happy New Year. So let's start. So today I thought of giving some glimpse of field relevant to your university and what I do. Use of NMR spectroscopy in drug discovery. So that's the topic that we'll be discussing. I will be really happy if you interrupt me at any time in point and ask questions if something is not clear. I hope my sound is clear to you, right?
Right.
Okay, fine. So what I do in our laboratory that we develop different NMR methods in solid as well as liquid, and we utilize those methods to understand many of the macromolecular complexes that actually like a ubiquitin E3 ligase and say amyloid fibers and nucleosome. Nucleosome is DNA packaging machinery. So we develop methods on one side, and then we apply those methods to understand these complex biomolecular assemblies. Now, if you look inside the cell, it's a crowded environment. So this picture has been generated by using the knowledge and research done by NMR, X-ray, and cryo-EM together. So it looks very crowded assembly, and there are many proteins, many organelles that are inside the cell. Now, in drug discovery, you choose one of them to target it. How to choose it and how to start, that's what the drug discovery program revolves around. So what pharmaceutical industry generally wants, they want to first identify a disease, which disease they are going to do drug discovery. So disease is specific drug. And then once they are identified, they have to synthesize those drug molecules and their analogs. Suppose we are working for a drug for pain. So drug like morphine has to be investigated, or say for malaria. If we are targeting malaria, then quinine has to be investigated. Initially, all these drugs will come from the plant product. When we understand the structural aspects of these drugs, then we can synthesize chemically these and their analog. Similarly, for heart failure. For heart failure, suppose we just 1 minute. This is an issue. Okay. So yeah, so digitoxin. So these are various drugs that are generally used. And once we understand this, one can synthesize an analog, like for say quinine, we have a HCQ, hydroxychloroquine. This can be scaled up, and then it can be formulated and sold and marketed. But initially, one has to identify a disease and target for that disease, and then you find a compound which is suitably working against that disease, and then one can scale it up. Okay, so that's start, and all these disease specific drug actually works something like this. So you have a protein molecule where these drugs will bind. So essentially these bindings is going to be a receptor small molecule interaction. So on receptor side, you have a specific site where the drug will bind, and this binding is mostly complementarity. So if we have a binding site which looks like a triangular shaped drug will bind, whereas this will not bind. Okay, so this kind of binding happens, and this is classically known in enzyme chemistry or even in the drug protein interaction is lock key mechanism. So where lock has a groove where a particular kind of key will fit and that open it up. Now in the second concept, which says there is a induced fit. So depending upon what is the structure here, but these molecules are not rigid, they are plastic, and that plasticity helps in adopting an appropriate conformation where drug and protein can interact. So this is lock key mechanism and induced fit mechanism. These are the two main hypothesized mechanism understood for drug discovery. What process it takes when we have a disease and when we have a protein? So we have to start with a discovery of the lead candidate which can bind. Then you have to develop that drug candidate. So that actually includes all of these target identification, where it is going to bind and how we are, what kind of drug that we are having, optimize that drug, and then we can go in preclinical stage, doing all the computational, biochemical, biophysical assay. Then it goes for clinical testing, like clinical phase 1, phase 2, phase 3, and final looking at this safety and efficacy, it is approved for market. So if you look at it, it starts from a target, a disease, and then development stage, then goes for animal testing. You check for like a ADME, absorption, toxicity, etc. Then you look on a healthy individual for looking at the safety and efficacy patients. Then you do registration, and finally, after years and years of work, like say 7 to 15 years, it gets marketed, it reaches to market. But if you look at it, it takes lots of money to develop a new drug, and that's how many times drug discovery program is really expensive program. So the drugs that reach to consumer is quite expensive because so much money has been invested, and of course the pharma industry wants to recover that money by selling this drug, and that's how many of these drugs are very expensive. So let's look at how we can discover a drug and where the structural biology tools can be of helpful, and I will specifically come to NMR spectroscopy. So say this is our drug, rapamycin. This, if you look at this molecule, if you look just carbon nitrogen structure, it looks something like this, but it has a three-dimensional structure where some portion is quite rigid and some are flexible like this. These are flexible, right? So if you just take drug as a rigid molecule, you cannot get the idea about flexibility. For this, you need to solve the structure. You have to get the XYZ coordinate of each of these atoms by solving the structure. Few of these moiety, which are especially methyl groups, are quite flexible. You look at their orientation is not fixed. Here the orientation are fixed. So this intrinsic dynamics that are associated with the drug molecule, they will have impact on binding to the receptor. So generally, it is required to solve the structure, and for solving the structure, we have a two prominent tool. One is X-ray, where you need to have a fine crystal, and then once you have a crystal, you can diffract and get a structure. The second one is NMR spectroscopy, which does not require crystalline order of these molecules, but it requires solubility. If they are soluble, you can do solution state NMR. If they are not soluble, one can do the solid state NMR, which can be done on a even microcrystalline, nanocrystalline, or powder form. Now, just to give you a glimpse of how X-ray works, actually you need to grow a crystal of the molecule that we want to solve the structure, diffract using X-ray beam, you get a typical diffraction pattern, and then from this diffraction pattern, one can get the high resolution structure of these molecules. So why it is important to have a structure? You can see if we solve a drug protein interaction and we can find it out what are the critical residue that binds to the protein molecule or receptor molecule. Here I am showing you two examples. One example where lots of hydrophilic or charged group are there, and you can see, sorry, here hydrophobic groups are there, here are hydrophilic groups are there. So if you look at it, this drug is surrounded by lots of hydrophobic amino acids like alanine, phenylalanine, valine, alanine, leucine. So here groups are mostly of like say aromatic group, here methyl group. These are hydrophobic groups. So hydrophobes will be supported by hydrophobes. Therefore, the binding pocket has a hydrophobic amino acids. And the precise orientation, one can get it if we have the high resolution structure. Similarly, if you look at this another drug, which if you look at here is O group, here O, N, and all those. So in this case, mostly the nearby residues are of hydrophilic nature or charged amino acid like arginine, aspartate, glutamate. So these residues are required to stabilize this drug. Now, the basic difference of drug binding pocket we are getting because we know what surrounds, and once we solve the structure, we know precisely how the drug binding pocket is and how drug is oriented in that pocket. So therefore, it is very important to solve a high resolution structure. You can see here, if the resolution of the structure increases, your side chain orientation also gets well defined. So at low resolution, you see methyl is flipping here, but in this case, it's get more oriented, and you can see we can define now better with the electron cloud here. We can define the orientation of this moiety in a better sense. So drug orientation at high resolution is very important, and that's what X-ray teaches us. Not only this, like a drug-bound protein structure for HIV1 protease, this drug called MVT101, one can see what residue surrounds here, and one can find the precise orientation of drug protein interaction. It not only gives the structure, it gives also the idea about the thermodynamics, the binding parameters. So like one can see here, this is a drug where we have a binding site, and here is a drug. Now to bind here, it has to repel all these hydrogens, right? And then this goes into bioactive conformation, it binds. So here first two things are happening, changing the water entropy and also changing its conformation. So one can get the thermodynamic parameter, and essentially the binding conformation changes. So one can get the thermodynamic parameter as well as the dynamics that is happening in the drug conformation. You can see in extended conformation, the energy is 0 kilojoule per mole, but in bound conformation, it is 10.5 kilojoule. So when it binds, it does not bind like a straight chain, it changes its conformation. So there is a delta G change in conformational free energy that is associated with the drug binding. So these structural insights also score for the biophysical parameter or thermodynamic parameter that are there. Now, again, another example, protein dynamics is very important during drug binding. So protein, as we discussed, it's not a rigid molecule, it's a plastic molecule. So when drug binds, there are lots of conformational change happens. And one can see here in the movie, I'll just show you here in the movie, you can see now when drug binds, there is a breathing motion and lots of conformational changes, especially here you can look at the yellow portion, it's changing a bit. Now, once we have a rigid kind of structural insight, this is not possible to capture such breathing motion that is happening in protein upon binding. So you need a tool that can understand the structure, the dynamics, thermodynamics together for a better drug binding interaction. And that's what NMR offers. Actually, it gives not only three-dimensional structure, but also dynamics that is happening in protein. So it gives idea about the fourth dimension in protein structure, which is thermodynamics. So if we look at the protein, there are all sorts of motion happening from picosecond time scale to second time scale, and one can classify in these two terms. One we can say as a local flexibility that is given by say how bond is vibrating, how methyl chain is rotating or loop motion, and there are collective motion where you can have a larger motion, larger amplitude motion, like a domain movement here and there. So all such motion, all the way from femtosecond time scale to second time scale, can be captured by the NMR spectroscopy. So to understand complete function of a protein, you need to have a structure, one need to have a dynamics, folding, thermodynamics, then only we can get the function. And there NMR plays a major role to capture this. So in Richardson's turn, because there is hardly any technique that is so informative and so many different type of application, and because there is no other techniques that provide so much fun. So not just information. If you start doing it, you also have of a pleasure of doing it. Now coming back a little bit of theory of NMR spectroscopy, I'm not sure how many of audience will be familiar with this. So I'll start with very basics. What NMR spectroscopy can investigate? So it can investigate essentially molecules that are like molecular size of different shape and size. It does not depend upon what is the order, like it does not require any crystalline order or any special sample preparation. Soluble protein, peptide, proteoliposome, powder, frozen solution, microcrystal, hydrogel, precipitates, aggregate, or biopolymer. Any of these kind of sample preparation can be investigated with NMR. Why? Because all of them has a nice thing, a nuclei, an NMR active nuclei. And if this nuclei is present in each of these, so then they can be NMR active and they can be investigated. Now, what actually we mean by NMR active nuclei? So a nucleus which has a spin quantum number not equal to 0, those are NMR active nuclei. And if we put those in magnetic field, essentially they will show some NMR activity. So what we are doing, these nuclei which are NMR active, or any compound or any preparation that we take where we have NMR active nuclei, we will put those in magnetic field. So as NMR has three terms: nucleus, nuclear, magnetic, resonance. So one has to have an NMR active nuclei, then we need to put in magnetic field, and then we have to establish some resonance conditions. So what we do, we take those NMR active nuclei, put those in magnetic field. So what happens upon putting it? So nuclei are randomly oriented here and there, and therefore net magnetic field, the net magnetic field because of this, net magnetization because of this is 0. But as soon as we put in magnetic field, say like 15 Tesla, they will orient either along the field or against the field, and therefore two levels will be generated here. Those two levels will have some energy difference, that is, say, delta E. Now upon putting those nuclei in magnetic field, we splitted that in two energy levels, so degeneracy in the energy level is lifted. What we need, an external RF source which matches with this. So if delta E matches with the H cross omega 0, then this resonance condition will be established. That means the nuclei from the ground state will go to excited state, and once they go, they will come back. So they will come back. So these nuclei will do precessional motion while coming back, and that generates a current. And this current is a time decaying function which is called free induction decay. And how they are doing precessional motion with a particular frequency that depends upon the nuclei. So omega H, omega C, omega D. That's a characteristic frequency for each of the nuclei, which can be plotted here. So this which will be encoded here in terms of FID. And when we do Fourier transform from time domain data to frequency domain data, we will get a particular resonance frequency that's for a particular nuclei. So that's what we do, probe the different nuclei in NMR spectroscopy. So just for an example, if we are taking this molecule carotenoidiester, we have different hydrogens, CH3 here, CH2 here, CH, and CH3. Now if we put all these hydrogens, since their chemical environment is different, they will give us different peaks in the NMR spectrum. So like this methyl will be giving the peak here, this methyl will be giving here, these CH2 is giving here, and these methylenes are giving here and here. Now different position, the peaks are coming at different position because they have a different chemical environment at the same magnetic field, and this term in NMR is called as chemical shift. Shifting in the resonance frequency because of differential chemical environment is called chemical shift. Now another parameter which is typically looked in NMR is called spin-spin coupling, and that determines how the peaks are splitted here. So that depends upon what is neighboring group. So this CH3 has a neighboring group here as a CH2. Now CH2 will split the CH3 into 3, 1 to 2 to 1. That's the typically we see here. Now say for this we are having neighboring group CH. Now the CH is splitting CH3 into two terms, 1 to 1, and so forth. So this is called spin-spin coupling splitting, and this is again a characteristic phenomena that we see in liquid state NMR spectroscopy. So chemical shift and spin-spin coupling are two basic building blocks of any NMR spectrum. Now, what happens when the molecular size increases? It is very difficult to get a decent spectrum. The spectrum becomes too crowded, and therefore we need to expand that in multiple dimensions. So multi-dimensional NMR essentially we do a series of 1D, but in another dimension we encode the frequency indirectly, like here. So we start with a preparation state, then we mix the magnetization, applying certain pulse or wait period, and then we detect it. And this separation between the preparation and mixing state we keep increasing. So here the another evolution state is created, and this is another time domain data that we have. So now in 2D we have two dimensions, T1 and T2, and once we do Fourier transform, we get a correlation peak which encodes two frequency omega-1 indirectly and omega-2 directly. So this is 1D, and if you do double Fourier transform, we are getting two-dimensional spectrum. So for an example, cyclosporine, we have here a crowded spectrum, and if we do two-dimensional spectrum here, like a proton-proton correlation, we can have now a beautiful, well-separated spectrum. So now this gives us idea how we can assign these peaks. So assigning means like what is where this CH is coming, where this CH3 is coming, and where CH2 is coming. So each of these peaks will have a position, and that's what we identify using resonance assignment. So using series of like a correlation spectrum, which can be proton-proton, proton-nitrogen, proton-carbon spectrum, we analyze this spectrum. So here one of these examples that I'm giving you, the proton-proton homonuclear correlation spectrum, and here heteronuclear correlation spectrum. Here is a carbon-proton. So in homonuclear toxi, you see these peaks, 1' is connected to 2', 3', 4' from 4' dash. So one can see here, here is a 1' connected to 2', 3', 4', 5'. And by just drawing a line, we can assign of each of these, and we do couple of time exercise, so we know that these peaks belongs to which one, and so on and so forth. But many times even proton-proton correlation is not sufficient, so you need to expand that using heteronuclear. So now we are establishing a carbon-proton correlation spectrum. So each of these has a proton, and we do two-dimensional NMR spectrum, where like here these three peaks coming from the methyl resonance, aromatic resonances. Here mostly comes from methyl resonances. So we can assign using couple of these experiments. Right. So after doing all these exercise, we finally can get the structure of an compound, small molecule compound. So essentially we are measuring chemical shift that we discussed. It's a very sensitive to local environment. We are measuring coupling constant that tells what is near what. So we can measure using through bond correlation spectrum or dihedral constant, and then we measure through space correlation here like NOE, and if required we can orient these molecules in a aligning media, and we can measure the dipolar coupling, and using all of these parameters, chemical shift, coupling constant, the NOE and dipolar constant, we can get the high resolution structure of a small molecule like we started with rapamycin. So if we have all these parameters, we can get the high resolution structure. Okay, now why this high resolution structure is important? Okay, so if you have any question, you please go ahead and ask, then I will continue from here. Any question? Dr. Nitin, is there any question up to this point?
Sir, no question till now. At the last the session, we asked the audience and we put the question in front of you. Sir. Thank you. So continue, please. Sir.
Yeah. Okay. Fine. So let's go ahead. Why we have to do so much of exercise to get a high resolution structure? You see, I'm just giving you an example of this thalidomide. This compound has a two conformation R and S. Now, this is used against morning sickness. So, you know, pregnant lady have a morning sickness. Now, initially, doctor were giving these drugs, and they subsequently they find that the childs are getting born, they have a like a birth defect, some defect has come. They wondered why this is happening, and then they found it out that if there is a S enantiomer of this compound, that is causing birth defect. Now you wanted to cure morning sickness, and because of side effect, you are getting a birth defect, and whole your future generation is getting hampered or grossly like a mismanaged because of such drug. So now they find it out, if just the conformation change, if you look at here, just configuration change is causing so much havoc. So it becomes important to go for an enantiomer drug, and that's what our modern pharma industry struggles with. Getting the like a polymorphism is really big headache for them. How to get the enantiomer drug is another issue that all the time they are looking. Similarly, like a ethambutol causes the blindness, and this causes the tuberculosis. So it should not happen that if you want to treat tuberculosis, you are making people blind. That's really going to be really dangerous. So therefore, getting absolute configuration of these molecules which you are taking, like you just imagine how much difference you are seeking. So just bond is rotated, RR changes to SS. That is going to create a serious problem. So we need to have enantiomer drug. And how to get this enantiomer drug? Like many times, if you crystallize, maybe your resolution is not good enough, you may not get it. And many times when you formulate drug and when you are synthesizing, whatever you are putting, when you put in formulation excipients, suppose conformation is changing, you don't know. So whatever is going in drug, you have to be absolutely sure, otherwise there is a problem. So one method in NMR has been developed, which is called residual dipolar coupling, where one can get unambiguous determination of conformation and relative configuration of a drug molecule. What we do here, so you know the J coupling that we discussed here, it's a splitting in the spectrum. Now this comes because say molecules, it comes depending upon what is near to what, like a CH2 near to CH3, then it will split into 3. Now if this molecule is tumbling freely inside, we have only J coupling. But if we restrict the tumbling, little bit restricted tumbling will reintroduce the dipolar coupling, and that dipolar coupling is not full, it's a partial, so D. Now this partial dipolar coupling gives us orientational effect, and that orientational effect can give us the relative configuration of the molecule. So essentially, you see our dipolar coupling in solution is average to 0 because all the spins are oriented randomly. But if we restrict it, there is a reintroduction of dipolar coupling, and that gives us relative configuration. So using such concept, one can get the absolute configuration of a drug molecule. Now that's all about the drug side. Now slowly I'll switch to protein side, how you determine the structure of a receptor or protein which is target for a drug. So generally protein structure determination involves 5 steps. What we do in protein structure determination, we have a primary structure and we have to go to tertiary structure or quaternary structure using NMR experiment. So what it involves, the steps taken in NMR for structure determination is first is sample preparation. You have to prepare a pure sample. After purity is ensured for sample, you have to collect the data which comes for months. Then you have to assign those peaks that I showed you using 2-dimensional and 3-dimensional spectrum. Then you generate the distance using distance like an OG spectrum. And after these are there, then you have to calculate the structure, and eventually you have to validate the structure using Ramachandran plot parameters. So Kurt Utrecht championed this and he got a Nobel Prize for protein structure determination. So essentially we bacterially express these proteins where we supplement glucose with 13C glucose and ammonium chloride with N15 chloride, and that's how we label the protein. It can be used also in a yeast culture or human culture, but those are more expensive. Bacterial culture is relatively cheaper. So one has to get really pure protein. So once we get a pure protein, you can check in SDS page or even in MALDI, then you can record a spectrum. So this kind of a spectrum is coming from protein, and you see how many resonances are there. It says just 76 amino acids. Too many resonances, and it becomes difficult to assign from the 1 dimension. So then we have to expand this into 3 dimension, and you can see now each peaks are separated. So these peaks are separated, that takes a week for recording, and then you have to assign it. So each of these dots should have a name which belongs to what. So after this determination, we measure the distances from each proton to other proton, and then using this distance, we determine the structure using simulated annealing. After this is done, we check the structure using ProCheck like a Ramachandran plot constraints, and that's validate our structure. So now we determine a structure for receptor and we determine a structure for a drug. Now how to understand the drug protein interactions? So you see, it's just molecular recognition. And the secret of life is molecular recognition. The ability of one molecule to recognize another through a weak bonding interaction, that's what is molecular recognition. So essentially the protein and ligand, how they are interacting, and in such interactions there are various forces. Those are intermolecular forces, so that determines the binding, and upon binding there is a conformational change, there is affinity associated, and upon binding it induces some response, which response is responsible for curing some disease. So what we monitor using drug protein interaction? So essentially some physical property we measure that exists between protein and ligand, and that's what we measure here. So generally ligand is a small molecule. What it has in NMR terms, it relaxes slowly, it will diffuse slowly, it diffuse faster, and it has positive NOE, and there is since it is a small molecule, so there will be negligible spin diffusion. Whereas a protein being a big molecule, it has a fast relaxation and it will diffuse slowly in solution. It has negative NOE and fast spin. When you form complex, now whole thing becomes bigger, and therefore it relaxes fast, diffuses slow, and then we will have negative NOE and fast spin diffusion. So we monitor these parameters. We can monitor on protein side and we can monitor on ligand side. So ligand side we measure by looking at their change in the chemical shift, or spin lattice relaxation, or even diffusion coefficient we measure. On the protein side, we measure the chemical shift SAR by NMR spectroscopy, or we look at how their HSQC is changing, 2D spectrum is changing, and that's what actually we measure. So one of the technique that I'm going to explain you, where we look at the ligand side and we understand the binding of protein and or receptor and the drug molecule. So see here my ligand, where I have a proton called H, and the chemical shift is at delta A, and here is my protein where proton has a chemical shift at delta B, and both of these happening in fast exchange. Okay, fast exchange means like I say micromolar range, so binding going off and all those. Now if that is the case, so if the binding is happening, we design an experiment where saturate or irradiate proton on the protein, and then if they are binding, there will be effect transferred here. If that effect is transferring here, that means they are interacting. If it is not transferring here, they are not interacting. So that's what experiment we are doing. So prerequisite for such binding, it should be in micromolar range, and also the ligand target ratio should be 15 times more. Now we irradiate on protein signal and look at the effect on the ligand signal. So ligand signal is reducing, and if we take difference here, if they are binding, we will have a positive signal here. If they are not binding, there will be not signal here. So this is typically we irradiate selectively on protein signal and look the effect of that irradiation on the ligand signal. So for an example, I'm just giving you. So here is my ligand molecule that was binding to protein. We did same experiment. We irradiated on protein and looked the effect on the ligand molecule. Now this is the reference spectrum where all the peaks are present, and when we did irradiation, now only few peaks are present here. You just look at few peaks are present, and these peaks are binding epitope to the receptor. So D, E, F, and B here. So only these moiety binds to the receptor, others does not bind. So we did not only get the binding, but also got information about where actually it is binding. So binding epitope is elucidated by doing this STD experiment. For an example, let's take two molecule, the 6-methyltryptophan and 7-methyltryptophan, right? We had a mixture of these two molecules. One only methyl group at here, and here is different. Now we had a mixture, we recorded this spectrum, and then we put the protein where it was binding, and we did STD NMR spectrum. So what we find after doing STD NMR, only few signals were there, and then we match the signal which was there. So here only matching, here matching, and here matching, and now we can find it out that all these belongs to 6-methyltryptophan and not to 7-methyltryptophan. So now we are sure that even within these two, a mixture, only 6-methyltryptophan is binding to the receptor, 7-methyl does not bind. Now that's what STD is giving us. To summarize, STD is a quicker method to screen the binders, and not only it gives the binding event, whether it is happening or not happening, it also gives the information about epitope that is binding. Okay, so that's what we do. So we take a mixture of compound, we irradiate on protein, and whatever is binding, it shows difference in the signal, and that's what is the binder. So STD NMR, it's a ligand-based NMR screening experiment to determine in a quantitative manner which compound binds to the protein in context of drug discovery. Ligand mapping, a more advanced use of NMR to direct characterization of protein ligand interaction at a molecular level through identification of important ligand moiety, and it also gives idea about binding constant called KD. Okay, so the other methods that we can use is the relaxation parameters. So like as we discussed, a small molecule will have a faster relaxation, and upon binding, the line width will change. So if there is binding, the lines become broader, and then also we know that whether this binding or not. So like here, we have a sharp line, and when it binds, it goes to broad line. So looking at the line shape of the small molecule, one can identify whether it is binding or not binding. So over the years, many experiment has been developed for ligand protein interaction, like a chemical shift differences in pharma methods, proton relaxation method, saturation transfer difference that I explained to you, transfer NOE or water log C, these all experiment enable us to understand the protein ligand interactions. So like just I will skip this. Now I am coming to what we do on protein side. So that was on ligand side. We go to protein side. Now for protein side, it is better to record a heteronuclear experiment. So here is my protein. We label these proteins with N15. We recorded the heteronuclear NMR spectrum, and then we titrated with the ligand. Now what happens, as you increase the concentration of ligand, few peaks start shifting. Here you can see it. You can zoom it here. So some of them are not shifting, some of them are shifting here. Now looking at the shift, we know that my ligand is binding at this position, like N20, G122, T125, and they are associated in a patch. They are located in a patch. So doing such experiment, like one can see here, these nice shifts are happening on few of these amino acids. So looking at these shifts, one can identify that what are again epitopes on protein or the binding site for on protein where ligand is binding. So this again enables us in ligand protein interaction. The other method is a transfer NOE, where we see the such kind of cross peaks, and then we can identify that binding happening. So there is no change if it does not bind, but there is a change in the sign of the cross peaks that says, like here you can see if the binding happening, change in the cross peaks, and this transfer NOE also helps us in identifying the ligand protein interaction. So if we know this, we can enable ourselves in doing the fragment based drug design. Suppose a protein has a very large binding site, so we can start looking at these binders, and different binders we can stitch together to have a covering all these binding site. Like here one can see it, finding it out, each of binders, stitching them together to have a complex molecule that will be more effective as a drug. Okay, so that was all the protein side. Now I'll just give you one example where solid state NMR was used to investigate an anti-cancer drug. So see, in cancer what happens? Uncontrolled cell division. And cell division is mediated by a assembly called microtubule. So over the years, so this what happens, microtubules bind to a kinetochore and then separates the DNA, and that's how this cell division is mediated. So any question?
Sir, one question is from the students. Please once again explain the reason of the fast exchange of ligand and protein to form complex of ligand and protein.
Fast relaxation or exchange?
The fast exchange of ligand and protein to form complex of ligand and protein.
Okay, okay. So okay, exchange here, whatever I
Yes, sir. And one.
I'll just come to that. So suppose
Yes.
I don't remember which slide I was, but anyway, I can take it here. So fast, like suppose these drugs are binding to the protein and going off, and this binding is of micromolar range. So that's not a very strong binding, right? So drugs should go, bind, act, and then get removed once the biological response is generated. That's we are saying kind of a fast exchange. Some ligand can go and bind permanently. Those are of very high affinity, like a nanomolar range. Binding and happening, this we are calling is a kind of relatively fast exchange. So that's what student means, or what?
One more question, sir.
Go ahead.
How ligand mapping is able to derive protein property of any compound?
How ligand mapping is?
Able to derive property of any compound.
Now, what property?
Physical property you have shown.
No, I have not shown any property that we are determining whether it is binding or not binding, doing
Okay.
such experiment. It will be good that a student can directly ask. No? Many times questions are not clear when they write it.
That is question from chat box, sir. Next time I will.
Many times I don't know what he mean. It could have been better if he just directly asked it.
Sir, you can continue.
Okay, fine. So I was discussing that cancer microtubule has been thought to be nice target because this plays a role in cell division. And over the years, people have come up with various drugs that targets microtubule, like paclitaxel. It binds to the negative end, and it's like a vinca alkaloid. It binds to positive end. So microtubule is a dynamic assembly. At one end it grows, in one end it just peel off. So if you destabilize any of these, you have achieved a drug, and that's what people have thought of doing the drug design for microtubule. So over the years, like a paclitaxel or ipothiolone has been investigated, which binds to the negative end. Now microtubule is made up of two tubulins, alpha tubulin and beta tubulin, and that's what has been targeted. So ipothiolone is again ipothiolone is another drug which binds to beta tubulin at the C termini, and then assembly is perturbed. So now people started solving the structure of tubulin bound ipothiolone, and they use electron crystallography, where they found that ipothiolone binds to a nice cavity. But the other group come up with solution state NMR, and they showed that the binding is differently oriented. Now here the question is that this was a fraudulent picture, and here it is quite flexible. Like in solution, it is flexible. They use in pharma method, so that was a little indirect method, and they gave a different orientation. If I just overlap these two, coming from NMR and coming from electron crystallography, you see the orientation of ipothiolone is very, very different. Now, what is actually correct orientation? This is very important question to be asked. So we sell out this experiment, and we thought of doing a more closer to the real scenario, where we took the ipothiolone. First, we solved the structure of ipothiolone using like a solid state NMR. We microcrystallize it, solve the structure, and then we dope this ipothiolone in microtubule, and we made a complex, and then again we did solid state NMR. So here you can see nice microtubule fibers, and here actually ipothiolone was doped somewhere in between. So we could record the signal of the drug molecule which was bound to the protein assembly, and we got a spectrum of this. And then we compared with a spectrum of free ipothiolone. We find that many of the peaks are shifting actually. So we measured this shift and then mapped on this structure, and we found that many residue, many actually moiety are showing a large chemical shift. So once we map it, what we saw that there are large chemical shift happening where none of these residue are in close contact when we compare with a cryo-electron microscopy picture. They are also not very well matching with the NMR spectrum. So we thought that probably none of these structure is 100% correct. There can be an intermediate way of binding, neither this nor this. So we propose an intermediate method, intermediate site where actually it could be binding with a different residue, and that's what using solid state NMR we found the drug protein bound interactions. Very briefly, in 5 minutes or so, I'll just show you how NMR can be applied in pharma industry. So already I gave you idea about competitive binding, like compound screening, the binding site location on protein. Then we can optimize how lead and structure elucidation. So all these are used in pharma industry, and those are done in biopharmaceutical. So biopharmaceutical, as you know, they can be virus, therapeutic serum, toxin, antitoxin, protein and peptide based drugs. They can be even monoclonal antibodies. So not only small molecule, but even larger molecule can be investigated with NMR. So like for a small molecule peptide based drug, You can do 2D NMR spectroscopy and you get a clear picture, and now you can even match with similar product and show that structurally they are similar. Similarly, one can investigate using like a methyl NMR spectrum of monoclonal antibody, which is 150 kilodalton. One can get a beautiful spectrum of maps. And then again, if you are going for biosimilarity, you can do the peak-wise peak similarity to establish how similar and dissimilar they are. So this I would like to conclude. NMR spectroscopy is an excellent tool for biopharmaceutical application. One can get idea about ligand and target-based experiments. Information about binding site and lead compound can be identified. Information about binding pocket can be figure it out. And one can show the biosimilarity for higher order structural analysis, and novel methods like a methyl trilogy, which I showed one slide back, can be used to look at the proteins up to 200 kilodalton. So with this, I would like to thank you all for having this talk. And then I would like to also thank my group members, the postdocs and staffs and PhD students that are working. Funding agency coming from different sources of Government of India and IIT Bombay for providing the facility. Thank you very much. Thank you.
Thank you, sir. I, Dr. Sachin Kumar, head of the department, IIMT College of Medical Sciences. On behalf of IIMT University, I would like to thanks to our eminent Professor Ashutosh Kumar for this knowledgeable presentation on NMR spectroscopy and drug discovery. I also thanks to our Dean sir to motivate us to organize this event. And I would like to thank all our faculty members and all participants to make successful this event. And definitely all of you gather the knowledge about the NMR and drug discovery. And again, I also thank to Professor Ashutosh sir to join us and give his precious time from his busy schedule. And I hope in future we will also share knowledge about some recent topics. Thank you, sir.
Thank you very much.
Thank you, Sachin sir. Thank you. Thank you, Ashutosh sir, for your valuable talk on the topic. So, sir, next time we expect that you will again join us for the next conference. And so, thank you, sir.
Thank you. Any other question? Or that's it?
Sir, that's it, sir. Thank you.
Okay. So I will then log off.
Yes, sir. You can.
Okay. Thank you.
सर आ गए लग रहा है मुझे हाँ गुड आफ्टरनून सर now I am going to start the next session by the Professor Javed Ali sir from Jamia Hamdard, and I would like to invite Dr. Vivek Verma. Dr. Vivek Verma is here. Okay. Now I welcome to Javed Ali sir. About the Javed Ali sir, Javed Ali sir passed his B.Pharm and M.Pharm with distinction from Jamia Hamdard in 1994 and 1996. and he earned his PhD in 2000. He was postdoctoral fellow at the Institute of Pharmaceutical Technology, University of Frankfurt, Germany, in 2005. Dr. Javed Ali sir has produced over 350 manuscripts in journal of repute and 5 Indian patents granted applied. He has an H index of 69 and more than 7,900 citations to his credit as per Google Scholar, and H index of 54 and more than 11,200 citations as per Scopus. He has featured as the world top 2% scientist, pharmacy and pharmacology from India on an independent study conducted by Stanford University, USA. Professor Ali is also actively engaged in committees of various central and state universities, Pharmacy Council of India, All India Council of Technical Education, National Board of Accreditation, Union State Public Service Commission, National Testing Agency, etc. He has been executive member of Indian Pharmaceutical Association Delhi branch from 2012 to date. He is a subject expert in pharmaceutical technology session of scientific session of Indian Pharmaceutical Congress held from time to time. So I would like to invite Dr. Javed Ali sir to deliver a talk on the relevant topic today's title of our conference. So welcome sir. Am I audible sir? Professor Javed Ali sir, are you connected? There is a unmute. Sir, am I audible? Unmute हो. Javed Ali sir, am I audible?
Hello, can
Yes,
you
sir.
hear me? I think there is a net connection issues, many a times I am, getting not connected and then I'm getting connected. I don't know. Maybe we will face some issues, but anyways, I will just start. Okay.
Okay, sir.
So at the outset, I would like to thank IIMT University Meerut, right? Particularly the Dr. Nitin who has invited me. I am thankful to the organizers as well for giving me the opportunity to interact with you all. And to start with, I am going to present my presentation.
Yes, sir. You can start, sir. Thank you, sir, for your acceptance.
Is it available? My presentation is available on the screen?
No, sir. Till now, not available. You can share, sir. Am I audible, sir? Presentation is not going. मल्टीपल शेयरिंग तो है ना इसमें? देना सर से शेयर नहीं हो रहा है ना, मल्टीपल शेयरिंग है ना?
मल्टीपल शेयरिंग शेयर कर सकते हैं सर।
आप मेरी आवाज सुन पा रहे हैं क्या?
Yeah, sir, right. And your slide is visible here.
I have made it, what I'm going to do is, in order to save this, I will keep my video off also, because
Okay.
otherwise there is a
Yeah,
net
right, sir.
connectivity issue.
Yeah, right,
Okay.
sir. Yeah, right, sir.
Okay.
Yeah, thank you.
Okay. So I'm going to talk about lipid nanocarriers, which is an emerging paradigm for improved therapeutic interventions. And my research group has worked on extensively on lipid nanocarriers. And the importance you can understand with the first COVID vaccine, which came into world for the delivery at by BioNTech Limited, is a lipid nanocarrier based vaccine. So you can understand the importance of lipid nanocarrier for various therapeutic interventions as such. Now coming to the lipid-based drug delivery system, when we initiated a lipidic-based drug delivery system in the early 1900 or 1850, we started with liquid emulsions, where we wanted to incorporate we wanted to incorporate the immiscible liquid with a miscible liquid and so on. I don't know if anyone can confirm that I am visible and the PPT is also going on, because I'm not getting any feedback. Hello? Can you confirm that the slide is going fine, or
Yes, sir.
there's
Yes,
an
sir.
issue?
Slide is going very fine, sir.
Okay, thank you.
Yes, sir.
In case there is a disturbance, please let me know so that accordingly I can take a
Definitely,
preventive
sure.
measure. Right? Okay, thank you. So liquid emulsions were there, and then it was thought that why not to incorporate drug in the liquid emulsions. The drugs which were water insoluble, they were solubilized in an oil phase, and they were dispersed in an aqueous phase with the help of emulsifying agents, right? But there were issues of stability and so on, and that's why we came with the from liquid emulsions to multiple emulsions, right? And then moving towards micro emulsions, then coming to nano emulsions and so on, including the micellar solubilization technique as well. But they were not quite, technique for a stabilized system. So slowly the scientists switched over from liquid emulsions to solid lipid nanoparticle, which are solid lipid matrices. And recently, nowadays, you will find a lot of nanostructural lipid carrier, which is a solid liquid with lipid hybrid matrices. And what was the reason for moving from liquid emulsions to solid lipid nanoparticle, also termed as SLN, to NLCs? The reason was nothing. It was talking about the stability and the issues of drug expulsion. Liquid emulsions, they constituted of liquid lipid matrices only, and they were having a high surfactant concentration. This surfactant concentration, which was acting as emulsifying agent, was leading to a physical instability, and there was a long-term storage. On long-term storage, there was post-wall ripening. Agglomerates were formed, which were leading to stability issues, right? However, the SLN which were formed, which were composed of solid lipid matrices only, these solid lipid matrices were having limited drug loading capacity because of a very defined structure. And on storage, there was a drug expulsion. So why not to bring irregularity? I think there's a background noise. If other speakers can be on mute, So this solid lipid nanoparticle had a very defined structure, a crystalline structure, which was leading to the stability issues, and there was expulsion also. So scientists thought of bringing irregularity in the structure so that it can accommodate more drug. and you can have a high drug loading also. And in addition to it, the problem of drug expulsion was also improved to a certain extent as such, right? So these NLCs came because of the improved drug incorporation and release properties, and a modulated drug delivery profile and stable drug incorporation during storage. So this was a journey how lipid nanocarriers become more useful, and finding usage in the use of the formulations cases for drugs which are having poor water solubility and having a high lipophilicity characters. The issues of concern with the conventional lipid-based formulation, whether they were liquid emulsions or microemulsions or nanoemulsion or solid lipid nanocarrier, was poor loading capacity. There was usage of good concentration of surfactant and co-solvent being used. And on storage, there was a possibility of many times drug precipitation, and that was also occurring when the system was getting encountered in the upper GI fluid. At that time also there was drug precipitation and the whole nano character was lost. There was expulsion of drug during storage, and there were certain toxicity issues as well because of the usage of surfactant when they were used at a higher concentration. So NLC is a novel approach of targeting drugs, right? This is a unique blend of lipids, right? Two types of lipid, a solid lipid and a liquid lipid. So liquid lipid brings about the irregularity in the very defined crystalline structure of solid lipid, and these are smarter generation of nanoparticle, right? And these matrices offer advantage of improved drug loading capacity and the release properties besides modulation of delivery profile. This concept was introduced by Mueller, a scientist in Berlin, right, in the year 2027. This is a schematic representation which talks about a SLN, which is having a perfect crystalline structure in nature, right? And the imperfect crystalline structure, which is in the case of NLC, because of the problem of the irregularities, they were able to get a good amount of drug being accommodated. When you are talking about major ingredients, I have already told you that there are two major ingredients in NLC. One is the solid lipid, and the another one is the liquid lipid. And in order to make them stabilize at a higher time period and a higher shelf life, so surfactant was also added in the formulation. We are going to take a case study also where we will talk about usage of surfactant and then how they were used. Advantages have already pointed out. They have higher drug loading capacity, improved drug release profile. They have stable drug incorporation during storage, and they also favor lymphatic uptake. We will see in the coming slide how oral delivery of NLCs favors lymphatic uptake as such. Applications are many: oral, topical, intranasal, parenteral, ocular, and pulmonary. Ocular spelling is single C. And primarily, we are going to talk about oral delivery, which is the most favored, patient acceptable, compliant route of administration, but we are going to touch upon other routes also. So parenteral application, you can protect drug from enzymatic degradation if you are giving the formulation parenterally, right? And you can have a high entrapment efficiency since you are using lipids of physiological nature, so they have a good biocompatibility also. It has also been found particularly for topical application because of the prolonged skin residence time. They have a small size. They will help in skin penetration because of the nano size, and they offer occlusive effect and improve skin targeting also. So for skin targeting, for localized delivery, they are the best, one of the best for type of formulation for application. For intranasal application, they bypass blood-brain barrier. They have bioacceptability, biodegradability, minimal toxicity, and the absence of burst effect as well. Ocular delivery, you can improve interaction with the ocular mucosa, and you can also prolong the retention time in the eye also, because of which there is a good amount of bioavailability which is being attained. Now coming to the core of the presentation, where we wanted to touch upon some of the case studies which we have done, particularly for oral delivery, where we have got a good bioavailability enhancement through nanolipid carrier approach. Oral drug delivery, the barriers are unstirred water layer lining the enterocyte side. So you already know as a student of biopharmaceutics, right? The unstirred water layer is a saturated layer, right? So there is a concept of sink condition. There is a concept of bulk layer concentration. There is a concept of saturation solubility. So the unstirred water layer is saturated with the concentration. So no more drugs come out and it is retained within the system only, right? And that's why you want to overcome the unstirred water layer. Secondly, the concept of P-glycoprotein mediated efflux and the intestinal metabolism by cytochrome P3A4, which causes the degradation of the drug in the intestinal mucosa. And secondly, the moment the drug is absorbed into systemic circulation, it is thrown out, it is effluxed out into the intestine through the ABC transporter, and one of the EPC transporter is P-glycoprotein. And the next is the first pass metabolism, pre-systemic metabolism, which drugs undergo, because of which you have to give a high concentration, high dose of drugs so that you can maintain the minimum effective concentration. So these are the major three barriers which we wanted to overcome through the formulation approach of nanolipid carriers as such. So these are physiological and biodegradable lipids which we are using, and they adhere to the gut wall, prolong residence time. They provide the reduced dosing frequency, because of which the drug release profile is sustained and many a time controlled as well. Minimize toxic effects on non-targeted sites, and the dose reduction is possible because you are able to get a high drug payload and able to maintain the drug release for a prolonged period of time. They have viable commercial potential because of the ease of scalability. Now, coming to the fate of drug encapsulated in the NLC after oral administration, the NLC is containing lipids, solid lipid and liquid lipid, and then the endogenous constituent in the lumen is a bile salt, which you already know that in the fatty meal. Particularly, very conventional example is a glycosylvine, which is given with a fatty meal, because glycosylvine is highly lipophilic in nature. So it is given with a fatty meal so that the bile salt which is secreted helps in the emulsification. Here also the lipids are mixed up with the endogenous constituent, that is the bile salt, forms micelles, higher concentration gradient for efficient diffusion across unstirred water layer. So this is the first coming the barrier. Second is a small particle size, has an improved surface area. They have an improved drug dissolution. There's a high dispersibility and effective adherence of the gut wall and prolonged intestinal residence time also. But what happens is that when you are using these micellar formation which are formed, they combine with phospholipids to form chylomicrons, which are generally taken up by the lymphatic system as such. The unique anatomy and physiology of lymphatics and the drug incorporated in NLC is the direct transport of drug through intestinal lymphatic into systemic circulation. So whole first pass effect is also obviated. Now you will say how to overcome the P-glycoprotein efflux. So the solution for that is that the lipid which we are using, they have the characteristic of P-glycoprotein substrate inhibitor, right? So they act as an inhibitor for these P-glycoprotein transporter. And similarly, if your lipid is also not showing any reported P-glycoprotein effect, inhibitory effect, then we can also incorporate number of agents which have been reported for P-glycoprotein inhibitors as such. So this is what I have talked about, the lipids mixed with bile salts, forming micelles. Hello, yeah? And then taken up by the system for the lymphatic vessels, which I have already explained. The drug candidates for NLC, as mentioned, they should be highly lipophilic in nature, preferably having a log P value of more than 4, and they should be, if they are a potential substrate of P-glycoprotein, so this is the ideal candidate because you can work very nicely to establish your rationale. The drugs candidates which are quickly eliminated, which are having a high, short half-life, right? So that they have a high elimination rate, suitable candidate for lymphatic transport, and which are extensively metabolized in the intestinal gut and liver by cytochrome P. The method of preparation of NLCs are many, but generally what we use in our lab is the high pressure homogenizer, right? Drug is dissolved, liquid melt is dispersed in surfactant solution, and the course pre-emulsion is subjected to high pressure homogenization at 100 to 1500 bars. The only problem working with this system is the drugs which are thermolabile in nature or which have the issues of certain heat related stability issues, then the problems comes into, but we have also found solution for that. So lipid degradation may occur at high temperature and pressure conditions, so we can have multiple cycle but at a lower pressure so that less heat generation is there. Extremely narrow size are formed, higher lipid particle content in the dispersion. There is an avoidance of any organic solvent, so there is no need for reporting any residual solvent at the last stage. Scale up and feasibility and possibility of wide range of production, that is also there here. The another method is formulation by microemulsion technique, where the organic phase in this the drug is and the lipids are dissolved, and an aqueous phase where the surfactant is mixed together, and with mechanical agitations they are there, and you formed an NLC suspension. Dispersion of warm oil in water microemulsion in a cold aqueous medium under mechanical stirring. High level of water content is difficult to remove, that is a problem, and then high concentration of surfactants is required, right? And there may be certain toxicity issues with the solvent and toxicity issues with the surfactant also, right? When the high pressure homogenizer is not feasible, you can always move for oil in water microemulsion. The another is the solvent emulsification, evaporation, or diffusion. So the liquid melt containing drug is dissolved in an organic solvent, which is emulsified in a water bath under mechanical stirring. And what is there in the water bath? An aqueous phase containing surfactant solution. The solvent is evaporated and the stable NLC dispersion is formed due to precipitation of lipid in the aqueous phase, right? There may be a toxicity issues with the solvent residues which are there in the organic solvent which you have used, right? And the particle concentration obtained using this technique is very low. Extremely small size of particles can be obtained, and there may be avoidance of heat during the preparation. So that these are the basic advantages, because of which this is also one of the preparation method for lipid nanocarriers as such. Then you can mix the high shear homogenization with ultrasonication. High pressure homogenizers, shear homogenizers are used, right? So melted lipid is dispersed in an aqueous surfactant, and then you use in a high shear homogenizer to do it. So you have high pressure homogenizer, you have a high shear homogenizer, you have a oil and water microemulsion, you have solvent emulsification technique, which are used for preparation also. So these are some of the reports which are there, which talks about using etoposide, vinpocetine, simvastatin being used in number of combination of a solid lipid with a liquid lipid with a surfactant like naturally one PGs and soil lecithin and so on. And then the reason the criteria of selection of drug was, one of the criteria was substrate for P-glycoprotein. They were having extensive hepatic metabolism and extremely metabolized in the intestinal gut as such. So this is another list which is there, and there are a number of papers which talks about the usage of NLCs, because and the many times increase in the transport, bioavailability, absorption, avoidance of P-glycoprotein efflux, and avoidance of first pass effect as well. Now this is the glimpse which is coming from the research work from our NPGR lab on NLC on tacrolimus. This is our ICMR. This was an ICMR funded project where tacrolimus, which is having a from the class of immunosuppressant, right? And it is a novel immunosuppressive agent for prevention the rejection of transplanted organs, challenge poor oral bioavailability and very poor water solubility, PGP flux, right? These are the criteria on the basis of which the drug was selected, and the strategy was the lipid nanocarriers as such. So for the design of tacrolimus loaded NLC, we screen liquid lipids to show the highest solubility of drug in the liquid lipid, which liquid lipid is having the highest solubility, and accordingly, we are screening the drug in the solid lipid, in which it is having the highest solubility. And then those selected solid lipid and the liquid lipids were seen for visibility, and then they were screened for the screening of surfactant by aqueous titration was done, and then finally going for NLC preparation as such. So these are the screening where the tacrolimus exhibited poor solubility in long chain triglyceride. However, it showed fairly good solubility in the medium chain triglyceride. So these are screening in the liquid lipids, and then solid lipid where it showed solubility in computerol 88880. Therefore, it was selected for a solid lipid as such. This was the screening of surfactant ratio to be seen, which was giving the highest emulsification area was selected. So these terminally phase diagrams were drawn, right, which was containing the solitol plus lecithin, right, and solitol HS15 alone, lecithin, and so on. And this crude emulsion area was selected, which is giving the highest concentration were used. So the final formulation, pre-formulation was containing oil and solid lipid, that is campiol, MCMC8, and computerol 88880, which was in the ratio of 2:3. And accordingly, the surfactant used was solutol HS15 and the lecithin preparation. The concentration range of lipid ranged from 1 to 2%, and concentration range of surfactant was 1 to 3% weight by weight. Since I have mentioned already that high pressure homogenizer causes the release of heat because of the high pressure, so the tacrolimus was having certain temperature issues, so we thought of bringing certain modification. So in a method A, it was a conventional solvent emulsification evaporation, as already talked about, and in which a probe sonication was also used for 5 to 7 minutes at 40% amplitude. What was the modification? The modification was the probe sonication was done only for 1 to 2 minutes at 20% amplitude. And accordingly, another method, which was high pressure homogenization, where conventionally we are using 10 cycles at 800 bars, but we modified the method for 3 cycles at much reduced pressure of 300 bars as such. So A, B, C, D, B and D are the modified method, and A and C are the conventional methods as such. So here in this, I have already explained that liquid phase and the aqueous phase are mixed together. The pre-emulsion is passed through the HPH system, right? And in the modified method, we are using the lesser, 300 bars only with 3 cycles to be produced. And here the same lipid surfactant ratio, total lipid, solitol, and the soy lecithin was used to prepare the NLC formulation. And these were the runs which we got from the various codes as such, and according to the size which we aimed, size up to 200 nanometer with a good value of mean PDI, polydispersity index, showing the homogeneity in the particle size, value less than 0.5, maximum it can go 1, and which was showing in a high percentage entrapment efficiency with the good amount of loading capacity, transmittance, viscosity. Accordingly, the formulation was selected, right? And from this data, on the basis of the particle size, mean PDI, entrapment efficiency, and the loading efficiency, we tried to prepare, got 2 formulation, right? And the method B was used, right? And method D was the modified method. So we came up with a modified method where we altered the method, and according to our requirement, we got the sizes and the entrapment efficiency of the formulation as such. So the preparation methods was much reduced sonication time of 2 minutes at 20% amplitude, which was the conventional emulsification method, and a high pressure homogenizer method was reduced 3 HPH cycles and 300 bars, right? Then the formulations were formed by 2 methods. They were taken for different characterization parameters, SEM and TEM. SEM for the surface topographic studies it was done, and TEM was done in order to correlate the particle size with the zeta size of which we obtained. And they were found to be, the shape was found to be smooth and textured as such. DSC results in the different formulation demonstrate complete encapsulation of drug inside NLC, in addition to the solid state of the NLC as such. So that was also being done. The FTI studies were done in order to find that the confirmed structural integrity of the drug was retained within the NLC, and that was also confirmed by FTIR results as such. Now, in order to study the in vitro drug release study, we simply used a SGF, SP, and SIF, SP. So why I'm talking about SP? Because SGF is like simulated gastric fluid without pepsin, and then the SIF-SP is the simulated intestinal fluid without pancreatine. So they were simple physiological buffers which were used to study in vitro drug release pattern as such. The drug incorporation in the imperfections of the binary lipid structure resulted in a greater amount of released drug as compared to drug suspension and the market formulation. And the comparison was done, and we got a higher percentage of drug release for a longer period of time as such. Coming to in vitro lipolysis studies, in order to find out what is the amount of the sediment and what is the amount of the drug in the aqueous phase, we just generally try to find out, do the in vitro lipolysis studies. So the higher amount of solubilized drug from NLCs in aqueous phase indicated that this much drug, which is more than 60%, would be available in solubilized form for absorption after oral administration, right? So this higher percentage of the aqueous phase was available as compared to the sediment, which reflects that you have a higher probability of drug absorption in that case. In vivo studies were done for doing the normal bioavailability studies in the blood, and the lymphatic distribution studies was also done in the animal studies as such. So the studies were done by HPLC MS method, and we just tried to find out that what is the amount of the relative viability. Also, the increment in relative viability of the nanostructured lipid carrier was found to be 6.2 and 2 folds, whereas for the NLC formulation, it was found to be 7.2 and 2.3 fold of that of the drug suspension and the marketed formulation. So in both cases, drug per se, drug suspension and the marketed formulation showed less bioavailability as compared to the nanostructured lipid carrier, which was ranging from 2 to 6 fold, 7 fold in terms of the relative bioavailability. This is the PK data, which talks about the Cmax values, Tmax, area under curve, 0 to T, 0 to infinity, AUMC, area under moment curve from 0 to T, 0 to infinity, T half key elimination, and you found, generally see that, you know, there is a 2-time increase in the Cmax value in the cases of NLCs as compared to drug suspension, and accordingly there is a greater extent of absorption. So the viability enhancement in terms of extent is almost like 10 times, 1079, and it is 171 and so on. And accordingly, it showed that it was a better opportunity to work in lipid nanocarrier formulation for the increase in the viability issues, particularly the drugs which are having a high lipophilicity character as such. A correlation between in vitro lipolysis and the oral viability results were also seen, that the formulation which was showing a higher level of drug available in the aqueous was more bioavailable, and accordingly it has shown a good correlation as well. This is the data on the biodistribution studies on organ and the limb distribution study as such, where how the drugs were distributed in heart, lung, liver, spleen, kidney, stomach, intestine, and lymph. The result demonstrated that the lymphatic distribution of nanolipid carrier was significantly enhanced, right, as compared to the tacrolimus suspension, which was used for the ANOVA analysis, and lymphatic uptake was greater as compared to the organ distribution in particular as such. So this is the summary which talks about that how the percentage of the tacrolimus was available in blood, lymph, and liver, and accordingly the nanostructured lipid carrier in comparison to drug suspension showed a better opportunity to work for the formulation benefits as such. The concentration of tacrolimus in blood and lymph was significantly improved, attributed to the medium chain triglyceride, long chain triglyceride based NLC matrix structural composition. The study demonstrated that NLC of tacrolimus is an extremely potential approach for enhancing its therapeutic prospect, and it should be explored commercially. So this was the outcome of the expert opinion, where we talked about immunosuppressive drug therapy, particularly the biopharmaceutical challenges, not only for immunosuppressive drug therapy, but also for NLC formulation. This was the outcome of the paper, which talked about the tacrolimus loaded nanostructure lipid carrier for oral delivery, optimization of production and characterization. This special issue was handled by Mueller, who is a pioneer in nanostructure lipid carrier, and he wrote a mail to me saying that you can submit a research paper, though it was a special issue on reviews to be compiled, but he was aware that we are working on NLC's formulation. So he invited us, we sent a paper, but he was so excited that he said this paper may be bifurcated. And the same issue, a paper came on the in vitro optimization of production and characterization, followed by tacrolimus loaded nanostructure lipid carriers for oral delivery in vivo bioavailability enhancement. So in the same issue, we have two papers on tacrolimus nanostructure lipid carrier, one talking about in vitro release patterns and optimizations, and then in vivo bioavailability enhancement, which I've just presented to you. This is another work in which we are not going into detail, but it is on the lipid nano carrier for isradipine, which is in HNF, Hamdard National Foundation supported project, where one of the students worked on isradipine, which has a problem, similar problems as that of the tacrolimus, with the issues of first pass, lipophilicity, P-glycoprotein substrate, and so on. And this came recently because this project was completed recently, and where the isradipine showed a better opportunity in terms of bioavailability enhancement when it was delivered to in the form of nanostructured lipid carriers as compared to suspension formulation as such. In another study, we also worked on neuropsychiatric drugs. There's a drug which is known as paliperidone, which is used for schizophrenia. And this lipid nano construct or lipid nano carriers were prepared for the formulation in particular. So till now, we have to, publishes in vitro work, and we are going to present get the in vivo work also being published in nanostructured lipid carrier formulation only, right? And the size was less than 200 nanometer, and a good percentage, up to 75% of cumulative drug release was also obtained. Confocal microscoping in small intestine shows a good amount of penetration as compared to the drug, which suspension, which was almost like 2 times enhanced penetration in the intestine. And the lipolysis studies also revealed that the paliperidone is having a good enhancement of drug solubilization when it is given in the form of a nanostructured lipid carrier as compared to the drug per se as such. Another study which is also being supported by ICMR, where we have prepared lipid nanocarriers for an anti-HIV agent, and we have got the result where the drug was compared with the drug per se in terms of the histopathological studies, in terms of the toxicity studies, and confocal microscopy in terms of the drug penetration in the intestine, in vitro lipolysis studies, in vitro permeation study, and the in vitro drug release profile of the drug as such. This is a study which we got published recently, where atrazanavir, which is also an anti-HIV, was given in the form of a nanostructured lipid carrier based approach for mitigating neuro-AIDS, thinking that the lymphatic system is also connected with the lymphatic system, and we thought of analyzing the drug levels in the brain also. And this is the published study where the nanostructured lipid carrier having a particle size of 227 nanometers was prepared. The in vitro characterization, confocal penetration in terms of with the use of a fluorescence dye was done in the brain region, and we just tried to find out that the Cmax in brain was higher as compared to the drug suspension which was delivered. It was almost like 3 times, and thus 3 to 3.5, which comes out to be 4-fold improvement in the brain delivery of atazanavir. So NLC is a potential approach for improving the drug bioavailability of highly lipophilic drugs with poor aqueous solubility. an extensive first-pass metabolism, and affinity for P-glycoprotein efflux transporters. In a way, high caliber of NLC system shadows the petition of other lipid-based dosage forms. Disrupted matrices of NLC consequently lead to higher drug loading. There is a higher drug entrapment, modulated drug release, and ultimately enhanced drug absorption as compared to other lipid-based formulation having uniform matrices of lipids. These special features of analysis are exclusively attributed to their unique composition, which is constituted of a blend of incompatible solid and a liquid lipid as such. Now coming to the future prospect, the promising results obtained using NLCs in animal models subject that the formulation should be extensively tested in human subjects. So there is a need for translational change from a lab studies or animal studies to the human studies, which is required. In vitro model studies, preclinical and clinical data on a wider range of drug candidates with differing physicochemical properties would help elucidate the mechanism of action of NLC formulations. And exploration of the potential of NLC formulations in improving the absorption of highly lipophilic drugs in conjunction with thorough understanding of the effect of formulation components, that is on efflux transporters, metabolic enzymes, lymphatic uptake, would help in completely addressing all the negative aspect of oral delivery of lipophilic drug as such. I acknowledge the collaboration with one of my colleagues, Professor Sanjala Babuta at Jamia Hamdard. I am also thankful for a number of my doctoral students who have come up with so many research outputs, and some of them, they are presently working, and some of them have worked on lipid nanocarrier and their formulation as such. And I also acknowledge the help and the research output of my PG or MPharm students for working in the lab and then coming up with the good results on nanostructural lipid carriers and the associated nanocarrier formulation as such. Thank you for your patience listening, and I will be happy to address any query or any question from your side. Thank you.
Thank you, sir. Thank you so much, sir. It was two interactive session. So, Verma, sir, questioner, please start and ask questions.
Good afternoon, sir.
Good afternoon, Vivek ji. Good afternoon.
Sir, basically, I'm having a question, not basically a one question, but it may be a kind of two questions.
Yeah.
So basically, when a researcher is starting with a research in some nanoparticles, so first step at which the researcher is facing a problem in a pseudo-ternary phase diagram, sir. So what kind of which software is very, I want to say it means is quite easy to understand just to draw a pseudo-ternary phase diagram, sir. Question first, sir. And question second is that, sir, among all those pseudo-ternary phase diagram, we are going to select a ternary phase diagram which is showing a maximum micro range or a nano range, sir. So basically, question is that, sir, how do we distinguish that the showing range is in micro range or in nano range, sir?
See, actually, ternary phase diagram is not going to give you the value for the micro or the nano range, number one, right?
Yes, sir.
Second is then what? Second is this is the this is a step in order to identify what concentration of the surfactant is miscible with your lipid phases, right?
Yes,
Thirdly is that, you know, once you have come up with the concentration of the surfactant which shows a good solubility of the lipids which you are working with, then finally you can get the size only through a zeta size or a time analysis. That is the way only, right?
Yes,
so maybe I think the more focused approach will be to use CCRD or BBD so that you can have the minimum runs of about 21 or 17 runs according to the criteria of the QTTP and QTP, and accordingly you have to select them as per your requirement. What is your aim? What type of particle size you aim for? What type of delivery route you are aiming? So accordingly, you can come up with that particle size, right?
Yes, sir. So, can you suggest one at least some software which is in terms of some easy to draw this pseudo-ternary phase diagram?
So generally the students are using the 3D graph papers for plotting the data, and then they show the maximum micro emulsion or the nano emulsion range, which we are talking about, right?
Yes.
But there are very few free software also available. If you Google them, that can also be used, where you can put directly on the chart paper and you can get the designated area. But we are doing it manually only, right? We are doing it manually only, and we are using it in order to only identify the S mix, right? If you, for example, S mix only in case if it is a formulation which is where we are using a surfactant or co-surfactant,
Yes,
but
sir.
here we are using a simple surfactant only. Okay.
Thank you,
Sir,
sir.
yes,
Thank
one
you.
more question from the chat box of YouTube Live. Sir, many more formulations are available here in the nano form. So why we will go for the NLC, nanostructured lipid carrier? Sir,
I have told let me answer this first. In the chat box, what I understand by the question is that you are saying that there are so many nano carriers which are available via NLC. I have already pointed out what are the potential candidate which can be taken for nanostructure lipid carrier, particularly the drug like which are having a high log P value of 4 or above. So they are very difficult, very strict molecule to work with, where you have a solubilization in issues and so on. So that drug candidate can be taken, number one. Secondly, the drugs which are having a high first pass effect, pre-systemic metabolism, right? Or the drugs which are degraded in the intestine by the mixed cytochrome system. So those drug candidates, for those drug candidates only I am recommending the nanostructure lipid carrier, because on the basis of which the drugs are having the specific characteristics, right?
Yeah. Thank you, sir. Thank you. Now hand over to Nitin, sir, please.
Thank you, sir, for your valuable talk on the nano formulation. Now, I would like to invite our HOD sir for give your welcome note. Thanks, vote of thanks for their presentation. Okay, sir.
Thank you, sir. I, Dr. Sachin Kumar, Head of the Department, IIMT College of Medical Sciences. On behalf of IIMT University, I would like to thanks to you for this wonderful and knowledgeable presentation on lipid nanocarrier. Thank you, sir. And I hope our all participants definitely benefited through this presentation, and they also gather the knowledge. And I would like also thanks to our Dean, sir, who motivate us to organize this event. And I also thanks to our faculty member, especially Nitin sir, Atul sir, and Vivek sir, to support us and help us. Thank you, sir, and thanks again. You join us from your busy schedule, and I hope in future you also bless us from your knowledge. Thank you, sir. Thank you very much.
Thank you, Dr. Saab. And I also thank the organizers, particularly the Dean, the head, Dr. Nitin, Dr. Vivek, Dr. Atul, right? So for arranging this international conference, inviting me, giving me the opportunity to interact. Thank you all. Thank you very much.
Thank you, sir.
So with your permission, I'm leaving now. Thank you. Thank you.
Thank you, sir. Thanks a lot.
Okay, thank you.
Now, wait for some minutes to join the next speaker with us, Dr. Mohammad Yasir. Wait for 5 minutes.
Okay, sir.
Yes, good afternoon, Ashish sir. Am I audible?
Yeah, you are audible.
Okay.
My voice is coming?
Yes, sir. Your voice is coming. And now I would like to invite Dr. Vivek Verma sir to introduce our next speaker, Dr. Mohammad Yasir sir from Ethiopia.
Thank you, Nitin sir. Am I audible, sir?
Yes, sir.
Yes, sir.
Good evening, sir.
Thank you, sir. Thank you for handing me this opportunity to get introduce our renowned speaker, that is Dr. Mohammad Yasir. Sir is having an academic experience of 14 years, presently working as an assistant professor, Department of Pharmacy, College of Health Science, Arshi University, Asela, Ethiopia. Sir has a professional qualification, PhD in pharmacy, and sir having an area of research in formulation and development, analysis of samples in biological matrices, lipid nanoparticles, bioavailability enhancement. And apart from this education and research, sir has undertaken a project that is being funded by AICT, 2 projects, presently projects funded by Arshi University, Asela, Ethiopia. And apart from this, sir is supervising M.Pharm students 10 and B.Pharm project supervised 22. Sir is having a publication 64, out of which 46 are research and 18 are review articles. Sir has a major publications elsewhere, Bentham Science, Taylor and Francis and Springles, etc. Sir has also published books 1 and chapters are 2. So, with all these details, I'm going to invite our speaker to please come forward and enlighten us with some topic knowledge. Nitin sir. Nitin sir.
Yes, sir. I think some network issues there.
There may be some. Sir, right now I have introduced sir. Is it audible or not, sir? Sachin sir.
Yasi sir.
Am I audible, Yasi sir?
Good afternoon, Yasi sir. Am I audible?
Yes, sir. From my side, you are audible.
Yasi sir is my colleague.
Achha.
I don't know he know me or not, but he is my M.Pharm colleague in ITA. Yes, yes, yes. And after a long time, I have seen him. Welcome, sir.
So it has been honored to invite Yasir sir as far as a friend of our head of the department. Please welcome. You are most welcome, sir. Yasir sir.
Sir, now you can start, sir. Thank you.
Yasir sir, there might be a problem with some mic or there will be a network issue. You are not audible, sir.
Hello, sir. Voice is coming.
Now you are audible, sir.
Yes, sir. Audible, sir.
Okay, sir. So the today's topic is pharmacovigilance of herbal medicines in Africa, present status and future prospects. You know, basically drugs are used for the treatment, medication, as well as diagnosis of certain disease in human being. The use of medicines is unavoidable. We cannot avoid the application of the medicines. The exposure of the certain medicines is started even before the birth, and this exposure increases in frequency and variety until the death. You see, the patient is taking the medicines for the treatment. If person is dying due to the somewhat a disease, it is acceptable and it cannot be avoided. But if the person is dying due to the use of medicines, it is unacceptable as well as unpredictable. But we know that no medicinal product is entirely and absolutely safe for all the people in all places at all the time. We must always live with some measures of uncertainty. So in this regard, WHO introduced a concept to see the efficacy, safety, detection, assessment, and understanding. Hello, sir. Hello. Hello.
Sir, continue, please continue.
Okay. So WHO introduced a concept which is known as pharmacovigilance. Pharmacovigilance is basically derived from the two words. One is pharmakon, it is Greek word which means medicinal substances, and vigila, it is a Latin word which means to keep watch. So pharmacovigilance is basically the science and activity which deals with the detection, assessment, understanding, and prevention of adverse drug effect or any other drug-related problems. This application this applies throughout the life cycle of medicines equally to the pre-approval stages as to the post-approval stages. Now we see the pharmacovigilance basically associated with the study of adverse drug reactions. This adverse drug reactions is nothing, it is the any response of a drug which are nauseous, unintended, including the lack of efficacy. So the pharmacovigilance concept it was introduced by the WHO to study the specifically the adverse drug reactions. It is considered as a worldwide master key for drug safety monitoring. WHO, FDA means US Food and Drug Administration, and European Medicine Agency, that is EMA, introduced legislation on pharmacovigilance to promote the good vigilance practices standards. Recently, its concerns have been widened to include herbal medicines, traditional and complementary medicines, blood product, biologicals, medical devices, and vaccines. Now we see what is the herbalism and pharmacovigilance of herbal medicines. The plant have been the basis for medical treatment through much of the human history, and such traditional medicine practice are still widely practices. Means from the development stages of the humans are using the certain medicines which are derived from the plants, and still nowadays the person are rely on the application of traditional medicines for their treatment. In this regard, the term herbalism as well as herbology and herbal medicines came into existence. It is nothing, this is the use of plant for the medicinal purpose and the study of such uses. If we talk about the herbal medicines include certain herbs, herbal materials, herbal preparation, and finished herbal product found in the pharmaceutical dosage form. As per the WHO, which estimates about the 80% population of the African countries presently use the herbal medicines for some aspects of primary healthcare, means majority of the population associated with the different countries of African region, they depend upon their treatment on herbal medicines. So to monitoring these events, the herbal vigilance concept came into existence. The herbal vigilance is the assessment of risk, benefit of herbal medicines and play a key role in the pharmacotherapeutic decision making. The ultimate aim of the herbal pharmacovigilance is to protect the patient from herb-induced harms and allow them to derive the maximum benefit from herbal medicines. Now we see what are the different aims of pharmacovigilance. The pharmacovigilance aim is basically associated with patient care, public health, risk benefit assessment, and communication. If you see towards the patient cares, the aim of the herbal pharmacovigilance is to improve the patient care, safety in relation to the herbal medicines, and all medical and paramedical intervention. Regarding to the patient health, to improve the public health and safety in relation to the use of herbal medicines. Risk benefit assessment means assessment of the benefit, harms, effectiveness, and risk of medicines, increasing their safe, rational, and more effective use. Communication to promote the understanding, education, clinical training in herbal pharmacovigilance, and effective communication to the public. Now see, the pharmacovigilance is basically a shared responsibility of companies which are developing the several pharmaceutical product. They should take the responsibility legally or morally because they are wholly responsible for development of the certain product. Regulatory authorities, they should take the position of pharmaceutical vigilance as they are giving the approval to the certain pharmaceutical companies. Doctors and pharmacists, nurses, which are responsible for taking the patient care. Stakeholder in pharmacovigilance, WHO, master stakeholder, government of individual countries, industries in a particular countries, clinicians, nurses, paramedics, academia, pharmacists, patient, and relatives. So according to the one study, in African zone, near about 2.5 to 18% of death of hospitalization is due to the adverse drug reaction. Before 2010, in African zone, ADRs were the four to fixed commonest cause of death in Africa. There are the certain reason behind this. First reason is associated with humanitarian concern, that is the insufficient evidence of the safety from preclinical trials and clinical trials of phase first to third studies prior to marketing. The authorization means before starting to the market, in case of especially in case of the herbal medicine, there is no big developed clinical concept as well as preclinical concept. It is generally standard that medicines are supposed to save the life. The person are taking the medication or patient are taking the medication for saving their life. If the some person is dying due to some disease, it is unavoidable. But if some person is dying due to the some drug, it is unacceptable or unpredictable. So we cannot expect that rather than a person is taking the medication for his health benefit, but rather than getting the health benefit, he or she will die. Another things, the cost of ADR is somewhat expensive. ADR related cost to the country exceed to the cost of the medicinal themselves. So conduction of the such type of the study, especially in case of the herbal medicine, is somewhat costly. Promoting the rational use of the medicines and adherence. Another reason is ensuring the public confidence regarding to the medicines, or another or last reason is an ethical practice. There are several persons who are selling the medicines for a particular use, but they knows well side effect or adverse effect of these reaction, but they are not telling to the patient. This is unethical. So unethical practice is another reason of such type of, you can say, the casualty in African zone or worldwide. So why we should conduct the herbal pharmacovigilance? What is the need of herbal pharmacovigilance? As the use of the herbal medicines has increased, so the report of suspected toxicity and adverse effect of herb should be there. We cannot say that herbs are obtained from the natural origin, so they are free from side effect. It is not happen usually. We can say the herbal medicines are traditionally considered harmless. It was the traditional concept, but nowadays they have the proper side effect. Since it is general faith that herbal medicines belong to the natural origins, so they do not have any side effect, but it is not true. They have the several side effect. There are the certain case report already published in the literature related to the adverse effect associated to the herbal medicines. The safety of herbal medicines has become an issue for the regulatory authorities, as serious effects have been reported, including liver, renal, cardiac, nephrotoxicity, genotoxicity, teratogenicity, as well as certain allergic reaction. So in this regard, WHO, recognizing the growing importance of herbal medicines worldwide, and they developed the guidelines for monitoring the herbal safety within the existing pharmacovigilance framework. Another reason for which the herbal pharmacovigilance is somewhat mandatory, it is the poor quality of the herbal material, incorrect, misidentified herbs, incorrect processing method, supplying of adulterated drug, contaminated drug, spurious drug, misbranded herbs and product lead to the undesired side effect, under effect of medicine, side effect of medicine, as well as adverse drug reaction. Reaction occurring as a result of overdose or over duration, tolerance, dependence, addiction, which can be detectable by conducting the pharmacodynamic as well as pharmacovigilance studies. Hypersensitivity reaction associated with the herbal medications, allergic reactions, idiosyncratic reactions, which can be detectable by pharmacovigilance, and mid-term or long-term toxic effect associated with the certain herbal medicines, which can be detected by in vitro and in vivo toxicological studies or by pharmacovigilance. Other reason due to which the herbal pharmacovigilance study should be conducted, it is medication error as that of the synthetic medication, lack of efficacy reports of herbal medicines, use of the medicines for indication that are not approved or for which there is an inadequate scientific basis, case report of adequate or chronic poisoning, assessment of herbal medicines related mortality, and abuse or misuse of herbal medicines, and certain drug interaction associated with the herbal medicine. Herbal medicine can interact to other herbal medicines. Herbal medicines can interact to the certain chemicals or synthetic drugs, or herbal medicines can interact to the certain foods. So these are the certain reasons. That's why we should conduct the herbal pharmacovigilance. The chronological development of pharmacovigilance. Around 1947, James Lynn reported the clinical trials showing effectiveness of lemon juice in prevention of the scurvy. 1937, sulfanilamide disaster was there. It was due to the some accidental, or you can say the some experimental issues. The drug was dissolved in diethylene glycol and given to the patient, and it was responsible to leading the death of more than 100 people because of renal failure. It was due to the toxicity of the diethylene glycol solvent. 1938, preclinical toxicity and pre-marketing clinical studies made mandatory by FDA before the application of the drug. 1950, aplastic anemia caused due to the use of chloramphenicol. 1960, the FDA started hospital-based drug monitoring program. The 1961, the idea of pharmacovigilance took shape mainly after thalidomide tragedy. It was given to the ladies who are pregnant, leading to the development of teratogenicity or fecomelia in the developed fetus. 1963, 16 World Health Assembly recognized the importance of rapid action on ADR, and British Committee on Safety of Drug Monitoring was established. 1964, in UK, yellow card system was started. 1964 to 65, National ADR monitoring system started in UK, Australia, New Zealand, Canada, West Germany, and Sweden. 1968, establishment of International Drug Monitoring Program by WHO. It was established in Geneva. And 1978, this center was transferred from Geneva to Uppsala. This is the brief history of, you can say, the chronological development of pharmacovigilance. Now if you see the status of the pharmacovigilance in contrast to the African countries, the establishment of the pharmacovigilance in African countries has lagged behind that of the developed countries. This is not a very old concept of development of pharmacovigilance in African countries. Prior to the year 2000, the pharmacovigilance was not prime concern to the African countries due to the several factors. These factors include the poor legislation for the medicines use, lack of access to the medicine and health commodities, weak and uncoordinated supply chain of medical product, lack of knowledge and awareness of pharmacovigilance, lack of financial, human, and technical resources. These are the certain reasons which are responsible for the, you can say, the improper, or you can say the lack of development of pharmacovigilance practices in African countries. In 1992, the center in coordination with WHO was developed in South Africa for the reporting of adverse drug reaction to WHO program for international drug monitoring. Followed by another center was developed in Morocco. So in 1992, two centers were developed in African zone, which are associated with reporting of adverse drug reaction to under the program of WHO International Drug Monitoring. In 1993, Tanzania and Tunisia developed drug monitoring program in their countries, followed by Zimbabwe in 1998. So up to 2000, only 5 African countries in the WHO program namely Morocco, South Africa, Tunisia, and Tanzania, Zimbabwe. 5 countries were there who are taking care about the some adverse drug reaction associated with the drugs. Between the 2002 to 2008, 10 other African countries participated and joined the PIDM program of WHO. And at the end of 2010, out of 104 full member countries of PIDM program, totally 24 countries associated with Africa were there. At the end of 2015, a total of 35 of 54 African countries were full members of PIDM. So it is increasing day by day, year to years. If you see at the end of 2017, total 123 countries associated with the WHO PIDM program. Among 123s, 39 were the African countries. So now we see the present status of healthcare system in Africa. Africa consists of 54 total countries with about more than 1 billion population, with an average gross national per capita income is around $1,100, with a diverse population of the people living below the poverty line. The healthcare system in African countries is somewhat weak, and healthcare professional is grossly inadequate. In a survey which was conducted in 2020, around 0.19 physician and 0.5 nurses and 0.25 pharmacists per thousand population were there. As per the capita expenditure on health in most of the African countries, it is below the it is below $100 as compared to UK, where it is 2,500, and in US where it is around 6,700. So expenditure on healthcare, it is very less in African countries as compared to the UK and USA. African shares the world pharmaceutical market. It is around 1.3% of total world pharmaceutical market as reported by the Pharmaceutical Research and Manufacturer of Africa. Due to the reason they have the less facilities, they are taking participate in the world pharmaceutical market. It is very less. It is around 1.3%. Due to this reason, 50 to 80% of the many of African countries have poor access of essential medicines. Pharmaceutical scenario in Africa is further complicated by the irrational use of medicines by both healthcare provider and consumer. Furthermore, there are genetic differences in the population, environment, and cultural dissimilarities which might impact on response to the medicines. So there are the certain regions which are responsible for the poor development or poor status of healthcare system in African countries. The present current status of pharmacovigilance in Africa, reporting of individual case report, that is ICSR, from Africa is extremely low as compared to the other part of the world. Means what is the rate of reporting individual case report to VG-base? VG-base is the database system of the WHO where the individual countries are reporting individual safety case report. If you see toward the African countries, it is around 0.88% as compared to the global numbers. So total reporting till date, it is around 1,03,499 as compared to the world, it is around 1,18,24,800. It is around 0.88% as compared to the global reporting. Now we see pan-African reporting countries. As in near about 54 African countries are there, and 30 among the 55, 39 are the member of WHO PIDM program. If you see toward the South Africa registered itself in 1992, and number of the ICSR case in VG-base up to 2018, it was around 28,000, and it covers about the 27% of African countries. Morocco joined in 1992. Up to 2008, they have reported 17,231. It is around 16.65% of as compared to the other, comparatively to the total African ICS reporting. Nigeria, they joined in 2005, near about 10,000 ICSR case they have reported to VG base. It is around 10.23% as compared to the total African ICS reporting. Like this, Egypt, Kenya, Tunisia, Congo, Ghana, Zimbabwe, and Algeria, they have joined to this database system in different years, and they have reported their reports to the VG base. Now we see overview pharmacovigilance of herbal program in Africa. If you see toward the herbal pharmacovigilance is still a very new concept, a very new activity in majority of the African country, except Morocco. Morocco have its own herbal pharmacovigilance program. It was created in 2004 along with the general pharmacovigilance program. Except Morocco, other countries set up this activity in last 10 years between 2009 to 2013. Among the 39 African countries, as we know, 39 countries are the members of WHO International Drug Monitoring Program. Only 16 countries have their own herbal medicine pharmacovigilance in coordination with their national vigilance centers. Some other countries have the herbal pharmacovigilance activities combined with the other services. They do not have separate pharmacovigilance program. Their pharmacovigilance program is usually integrated with other services like Drug Information Center, Pharmacovigilance of Other Health Product, Poison Control Center, Authority of Registration and Evaluation of Herbal Medicines. So among the total 54 countries in African continental, 39 countries are the part of WHO International Drug Monitoring Program. Among the 30, 16 countries have their own herbal pharmacovigilance in coordination with their national pharmacovigilance program. Besides this, other countries do not have the separate pharmacovigilance program, so they are doing their pharmacovigilance activity along with herbal pharmacovigilance activity in coordination with other centers like Drug Information, Pharmacovigilance of Other Health Product, Poison Control Center, and Authority of Registration and Evaluation of Herbal Medicines. Now we see toward the date of setting up of herbal pharmacovigilance activity and herbal pharmacovigilance organization system in certain African countries. If you talk about the Angola, they joined the pharmacovigilance activity in 2009, but still they do not have separate pharmacovigilance center. They are working on some pharmacovigilance activity along with the Drug Information Center. Botswana, they established their pharmacovigilance activity in 2004. They have the one national center for reporting the pharmacovigilance activity. In this, along with other pharmacovigilance activity, they are doing some herbal pharmacovigilance. Now, other countries include, that is Burkina. They joined in 2009. They have the network with sub national as well as regional centers, or they are doing the pharmacovigilance activity along with the herbal pharmacovigilance with Drug Information as well as other vigilance. Democratic Republic of Congo, it was established in 2009. They have the National Pharmacovigilance Center. They are doing some vigilance activity along with Drug Information and rational use of the drug. Egypt, Ghana. So like this, all 39 countries out of 54, they have the somewhat pharmacovigilance activity, either this activity running under their well-developed pharmacovigilance center, or if the pharmacovigilance center is not there, so such type of the activity they are coordinating under or in coordination with other type of the centers. Now you see number of herbal medicines case reported in different countries associated with Africa. If we talk about the Angola is still reported around totally 5 herbal pharmacovigilance associated activities. Among the 5, one reported in 2018. Botswana, they have reported to Vigiflow only one activity, means one herbal medication, one herbal associated adverse drug reaction they have reported to Vigiflow. Burkina, they do not have the well-developed system. They are doing all these activity manually. Only 4 activities still reported. Among the 4, one is reported in 2018. Democratic Republic of Congo. They have reported this pharmacovigilance activity to directly to Vigiflow. Total 18 activity they have reported. Among the 18, 3 they were reported in 2018. Egypt, they have the manual system for reporting the ADR. Ghana, they have the Vigiflow. Liberia, Mauritius, Morocco, and Nigeria. So some countries have well-developed pharmacovigilance activity. So they are they are observing the pharmacovigilance or ADR associated with different herbal medicines, and they are directly reporting to their pharmacovigilance center. And through the pharmacovigilance center, these activity are reporting to the Vigiflow, a database of WHO. Some country have still not well developed. They have the manual facilities, so they are reporting through the manual sources. So these are the other countries and their total case report. Now, regulatory bodies of pharmacovigilance in various African countries. 39 African countries belong to the WHO International Drug Monitoring Program. All countries have their national pharmacovigilance center independently or associated with others. If they do not have independent pharmacovigilance center, their pharmacovigilance activity is coordinated with other centers. So totally 18 centers of national, totally 18 among the all countries, total 18 countries of national center are affiliated with the Ministry of Health. Means they have the well-established center, or they are working under the supervision of Ministry of Health. And other countries, their pharmacovigilance activity is running under other drug regulatory agencies. If you talk about the South Africa, in South Africa, in 1987, National Adverse Drug Event Monitoring Centers, that is NADEMC, was established. It is a unit of Medicinal Control Council, South Africa. It was established to facilitate the adverse drug reaction monitoring. The NADEMC managed the collection and reviewing of voluntarily submitted ADR reports from the healthcare professionals to detect the signals of unknown or properly understood or poorly understood ADRs. South Africa became the first African member of WHO International Drug Monitoring Program in 1992. The pharmacovigilance activity are conducted in National Department of Health under the Ministry of Health. In South Africa, pharmacovigilance activity initiatives are largely driven by the three key stakeholders. One is regulators and pharmaceutical industries. These type of the stakeholder basically focused on product. Second stakeholder is public health program. These are generally focused on the system, means the drug system, or the other stakeholder is healthcare providers and clinicians. They are usually focused on patient. If you talk about the Zimbabwe, the National Pharmacovigilance Centers was set up in 1998 by the Medicines Control Authority of Zimbabwe in collaboration with University of the Zimbabwe, medical schools, and associated with Department of Pharmacy, Department of Drug and Toxicological Information Services, and Ministry of Health and Child Care. If you talk about the Kenya, pharmacovigilance is regulated by the Pharmacy and Poison Board. There is no separate pharmacovigilance authority. The pharmacovigilance activity are running under Pharmacy and Poison Board. The pharmacovigilance program is supported by the School of Pharmacy, University of Nairobi, through its Master of Pharmacy program in Epidemiology and Pharmacovigilance. University of Kenya, Nairobi, they are running the M.Pharm in pharmacoepidemiology and pharmacovigilance. If you are talking about the Ethiopia, the Food, Medicines and Healthcare Authority of Ethiopia is responsible for pharmacovigilance activity. The pharmacovigilance program is supported by the School of Medical Science, Addis Ababa University, Ethiopia. So beside these countries, every countries have their own body of pharmacovigilance, which is running under the supervision of Ministry of Health or other regulatory bodies. Like if you talk about the Angola, the pharmacovigilance department is there, which is running under the Ministry of Health. If you talk about the Botswana, there is a drug regulatory unit. Under the drug regulatory unit, the pharmacovigilance program is going on. It is running under the Ministry of Health. It is Kenya. Directorate General of Pharmacy and Drug Laboratory is there, under which the pharmacovigilance program is running, and it is coming under the Ministry of Health. Now we see other countries, near about 18 countries are there which they have their pharmacovigilance activity directly through the pharmacovigilance center or through the some regulatory bodies, and these regulatory bodies are running under the Ministry of Health. Now we see other countries, their pharmacovigilance activity, or you can say the regulatory authorities which are governing the pharmacovigilance activity, they are running except Ministry of Health. They have the other bodies. If you talk about the Cape Verde, the Food and Pharmaceutical Product Regulatory Agencies is there, under which the pharmacovigilance department is running. Egypt, Drug Regulatory Authorities is there. Under Drug Regulatory Authorities, the Egypt Pharmaceutical Vigilance Center is there. Ghana, Food and Drug Authority. Under the Food and Drug Authority, Safe Drug Monitoring Program is going on. Liberia, Medicines and Health Product Regulatory Authority is there. Under this authority, pharmacovigilance and medicinal information are running. So like this, many all 39 countries are running their pharmacovigilance under the direct supervision of pharmacovigilance center or under the supervision of other bodies. Challenging in monitoring of safety of herbal medicines in Africa. What are the different challenges? In African countries, small research programs are going on. We see the much research have been conducted on traditional herb medicines in African zones, but a very little are clinically acceptable, because the research are conducting at laboratory stages. From this laboratory studies, they are not going for preclinical as well as clinical trials. If you take the example, near about 1,200 plant species have been reported for the treatment of malaria, but only 13 undergo for clinical trials. So small research are conducted at laboratory scale. From that laboratory scale, these studies are not going for preclinical as well as clinical trials. If very small studies have been forwarded. The reason behind it, there are certain difficulties. These difficulty includes resource constant, means it includes the lack of funding, equipment, staff, as well as improper infrastructure, lack of infrastructure. Another reason is social elements, including the difficulty in recruiting enough patients, poor report from traditional healers, and lack of vigilance of biomedical staff to be involved. Another reason for improper development of the vigilance, it is the poor herbal medicine supply, including insufficient cultivation, production, and quality control, lack of trained staff, logistic issues in conducting trials, regulatory framework are underdeveloped or not developed in most of the African countries. Other reasons are the poor quality of the drug and poor quality assurance. Unlike the synthetic medicines, as we know, the herbal medicines are chemically rich. These are complex and not present as a single compound. In a particular herb, you can say even a single active principle will be there. There is a lot of active principles may be there. This make more difficult to ensure as well as to maintain the quality of a drug. Furthermore, there are certain problems associated with the herbal product. It includes herbal drug interaction, unknown composition, lack of standardization for the dose of active component, inappropriate dosing of herbal medicines, lack of proper development of pharmaceutical dosage form, contamination during the harvesting of herbal product, improper labeling. These are the certain reasons which make the system further complicated. Furthermore, a number of the factors that can influence the quality as well as quantity of chemical constituent present in the herbal medication include geographical origins like climatic, soil and photoperiods, genotypic variations, and part of the plant which should be collected that have the much active principles like leaves, stem, root bark, harvesting time, maybe it is around the year, it may be the seasonal time of the day and conditions, storage and processing and extraction, complications, and combination of herb and processing with combination of herbs and or processing of combined herbs as the medicines. As we know, the standardization regarding to the herbal medicines are there, but standardization of herbal related active principle individually, it is really performed, because we are not performing a proper standardization for individual active principle which is present in a particular kind of the natural plant. So this leads the intrinsic variability result in the product that may be the quite different and not necessarily bioequivalent even when from the same single herbal ingredient. So how to monitor and report the safety of herbal medicines? A general procedure is there. The safety of herbal medicines can be monitored through a national pharmacovigilance center. A pharmacovigilance system is the network between the all stakeholders, including national, regional pharmacovigilance center, health facilities. It includes both private as well as government facilities, drug stores, pharmacies, patient, consumers, and manufacturer. So depending upon the availability of all stakeholder, there is a network. Through the network, any problem associated with the herbal medicines, they can report to their regional centers. So general method is there. So if any problem is coming through the herbal medicines, or you can say the synthetic medicine, first of all the stakeholder will report to the regional pharmacovigilance centers, or this regional pharmacovigilance center will evaluate. If it is okay, then they will report to the national pharmacovigilance center. This national pharmacovigilance center, through the pharmacovigilance committee, they will evaluate these complications or complaint, and if it is okay, they will report this thing to Uppsala monitoring center. Now we see WHO traditional herbal medicines program in Africa. To enhance the expansion of herbal medicines, the World Health Assembly WHO passed a number of resolutions, and in 2002 a comprehensive traditional strategy was published by WHO which focus on the 4 areas to improve the national policies and regulation in African zones with integration of in integration with traditional medicine practice within the national health system. Second is safety and efficacy and quality of the herbal medicines. Standardization means standard for ensuring the safety, efficacy, and quality of the herbal product. Registration of traditional medical practice providers, so that they can ensure the safety and quality and efficacy of the herbal medicines. Accessibility, official recognition of traditional medical practice provider to ensure the proper accessibility, and last one, to improve the regional use of herbal medicines. Now in this regard, 2007 the Traditional Practitioner Act was enacted. In 2008 a draft associated with the national policies on African traditional medicine was published. To facilitate the evaluation of the traditional medicine in African zone, WHO Regional Office Africa organized a first regional workshop on regulation of the traditional medicines. It was conducted in Johannesburg, April 2003. The workshop was attended by 18 national drug regulatory authorities and experts. To promoting the registration and marketing of safe and effective and good quality traditional medical practices, the second regional workshop was conducted under the supervision of WHO. It was conducted in Spain, and total 15 national drug regulatory authorities associated with the African countries were attended this workshop. In addition, WHO Traditional Medicines Strategy 2014 to 2023 aims to support member state in developing proactive policies and implementing the action plan to strengthen the role of traditional medicines. Now we see what are the different adverse effects associated with the some herbs which are growing in African countries. If you see the Prunus africana, it is useful for the treatment of the prostate cancer, but it has a potential adverse effect associated with the gastrointestinal upset. It is the product of South Africa. Violatory, it is used for nervous system ailments, fever related to malaria, dysmenorrhea, and abortion induction. Basically, the potential toxicity associated with this herb is acute kidney injury, diarrhea, and dehydration. It is obtained from the Zimbabwe and Sudan. Another is Catharanthus roseus. It is used for the treatment of cancer chemotherapy. And the potential side effect associated of this herb is incoordination movement. And it is the product of Madagascar. Hodia cactus, it is appetite suppressant. And major adverse effect is hyperglycemia. It is the product of South Africa and Namibia. That is Spanish. It is used in dialysis to ensure the major potential adverse effect of this drug is extensive organ and tissue damage with the death induction. It is obtained from the Ethiopia. Bird flower, it is used for the treatment of the dysmenorrhea. The potential adverse effect is nephrotoxicity, is obtained from Zimbabwe. Impala, it is used for impotence, gastrointestinal ailments. The major potential adverse effect is chronic renal disease. It is obtained from South Africa. CART, that is Catha idilis. It is used for the central stimulant action, and the major adverse effect is hepatotoxicity and cardiotoxicity. It is obtained from East Africa. Licorice, that is Glycyrrhiza glabra. It is used in sore throat and cough, and major potential adverse effect are amenorrhea and heart failure. It is obtained from Morocco. Now, these are the certain herbs which are obtained from the certain African countries. They have the potential application as well as potential adverse effect. So before the application, a study should be conducted to ensure their effectiveness as well as to ensure their potential drawbacks. Now, the nature prospect and activities associated or should be conducted in African zone to improve the pharmacovigilance system. The recommended approaches is to include the herbal medicines in the existing national pharmacovigilance system where such system have not been yet developed. Means if some country have the proper pharmacovigilance system but they are not conducting the herbal pharmacovigilance, they should conduct, or they should include the herbal pharmacovigilance in their existing national pharmacovigilance program to establish or improve the comprehensive national pharmacovigilance system with coverage of herbal medicines. Prepare the guideline to identify the particular challenges faced in monitoring the safety of herbal medicines effectively and propose the approach for overcoming them. Special attention should be given to improve the reporting the HM based ADR and analyze the cause of reporting ADRs. Support for the development of a specialized pharmacovigilance tool related to the herbal medicines. Improve the manpower, including technical staff separately for herbal pharmacovigilance program. Strengthen the existing regulatory system, if any, or create a proper regulatory system associated with the herbal pharmacovigilance. Now, the exact system of the herbal pharmacovigilance is very poor in major of African countries. Continuous training programs should be given to the traditional practitioner to increase the awareness of herbal medicine, ADR, and reporting system. Scientific evidence of safety, efficacy, and quality of the herbal medicine in most of the countries should be improved. A very few academic institutions offering any formal education associated with the pharmacovigilance based on herbal medicines, it should be incorporated in the national curriculum. Work should be done to improve the communication between the health professional, consumer, other stakeholders, including the regulatory authorities, suppliers of herbal medicines, to increase the awareness of herbal ADR. And this is the last but not the least. Country usually integrated the pharmacovigilance of herbal medicine into the existing pharmacovigilance system, and they are working as that of the same manner, but it should not be happening. In fact, the duplication of the system is not very useful. A separate pharmacovigilance system should be developed especially for herbal medicines. So this is all about the herbal pharmacovigilance in African countries.
Thank you, Yashi sir, for your valuable speech on the topic African pharmacovigilance, herbal pharmacovigilance. And I give you special thanks for your availability and your presentation. from your precious time of your busy schedule. So I hope
Thank
you
you, sir.
you will share your knowledge with me and with us in future also. Okay.
Sure, sir. Sure, sir. Welcome. If you give the another opportunity, I will do the welcome.
So I would like to give thanks to our Vice Chancellor sir, Pro VC sir, and Dr. Satish Bansal sir, and our Dean sir, Dr. Anuj Mittal, and HOD Dr. Sachin Kumar, and all faculty members for being present here, all the students who are benefited today, 3 different sessions delivered by the 3 speakers. So for the next proceeding of the conference, next day we will be available on the same time, 11 a.m. on 4th January. So thank you. Yashi sir,
Sir.
you may leave, and
What,
so
sir?
again, thanks for your presentation.
Thank you very much, sir. Thank you very much. So,
Yes, sir, it was expected that we will meet soon again.
sure, sir. Sure. Sure, sir.
Thank you, sir.
available.
Thank you for
I'm,
your time, sir.
I am available because Dr. Nitin, we work together, and Dr. Sachin, we are the batchmate. He is doing M.Pharm in pharmaceutical chemistry, and I was doing in pharmaceutics. Even Dr. Anuj Mittal is my batchmate. He's senior, but we completed our M.Pharm together.
Acha, acha.
So all we, you can say, the colleagues. So if you will give the further opportunity, I will accept happily without any
Exactly,
problem.
exactly, sir. With a hope, we will meet again. Once again, thank you, sir. Thanks for your
Thank
help.
you, thank you very much. So, can I leave, sir?
Yes, sir. You can leave, sir.
Okay, sir. Thank you, sir. Thank you, sir.
Okay.
विवेक सर ये कैसे करना है फिर इसको
There was
मैं
an
वो कर दूं
option.
इसको
There was an option. First, you conclude with this Zoom app. Sir.
ऑप्शन
Just turn it off. Zoom app, sir. Zoom app turn off कर दीजिए.